And for our last speaker for today, Dr. Guy Shtar from Ben-Gurion University. So no one can claim that we only take Tel Aviv University here. And Guy will talk about crossing boundaries, a machine learning journey from antibacterial agents to cancer therapy. Please.
Congratulations on making it to the final talk. You made it. I am Guy Shtar. Today I'm a data scientist at Intuit, but I'm going to tell you about a research I've conducted as part of my PhD in Ben-Gurion University with great people. You can see their names here. I'll take you to a short journey and tell you how we took an antibiotic from the shelf in the pharmacy and show that it has anti-cancer activity. So the real goal was to develop a system and not to just focus on one drug. This was funded by the Israeli Innovation Authority, and we were after a system that can repurpose drugs, approved drugs. Repurposing means taking a drug from the pharmacy, approved, and show that it has activity that no one knew about. And why would we want to do that? First, because it's very hard to bring a drug to market. It costs a lot of money, it takes a lot of time, the safety profile is unknown in the beginning, and it is prone to failure many times. And we're not the first one that do repurpose with AI. Many did before us, but we did some things differently. We used different modalities. You'll see here, I'll talk about graphs and tabular data and different types of features. We use recommendation systems methods. This is something we had experience with, and it helped us here. Here, we use domain experts. Pharmacologists actually took part in labeling the drugs, the data, and looking at the results of the model. And finally, probably the most important thing we did, I'm an expert in AI, not in biology. So when I saw the problem, I said, this is a binary problem. Cancer, yes or no? This is not the go-to solution in biology. Most of you probably know. You should focus on targets. We didn't focus on targets, but we finally look at them, not as the main goal. And this was important and not straightforward. The methods are as follows. First, you need to collect data. So we collected a list of drugs and split them into anti-cancer, yes or no. You can see the sources of these drugs here. There are a bunch of data sets. By the way, the negative list was harder than the positive one because you cannot prove that the drug is not anti-cancer. There are many types of cancers, and no one really tries to prove this. So we had some heuristics to collect the list. And then we did some feature engineering. So we focused on small molecules, and we knew something about the drugs even without collecting these features. But drug targets, as we said, are very important as you start. And then we also worked with drug-drug interactions, different types of interactions that we had a lot of experience with. This is actually how I started my PhD. And we tried more features, but these are the ones that were really important finally. And when it comes to modeling, I had this fantasy of working on a new neural network or something at the time, but to get into reality, we had 1 year to do everything I show you here in this slide. It's no time, and when we started working on that, a few months before, a group of researchers from MIT, by the way, led by an ex-Israeli, Regina Barzilai, they published a neural network with the code and everything called ChemProp that can do a prediction of activity of molecules. So we took this neural network and we extended it with the data that you see here. And not only did the team from MIT develop this neural network, they even discovered a new antibiotic, something that wasn't done for years with this neural network. So this was a good motivation for us not to develop anything new here, but to focus on the rest. And I said that we did not start with the targets, but we wanted some picture, something to know about the potential activity. So we had another neural network that can predict, given a molecule, which targets it activates, and I'll show a little bit more about it later. And then, of course, we took a candidate drug, we moved with it into the laboratory, and we had results. You'll see the process now. First of all, in silico results, so this is the binary model, the one that predicts cancer yes or no. The first line here is the baseline. It means no features, just the structure, the atom and the bonds of the molecule. It worked relatively well, but of course we didn't stop there. Second line is adding some feature engineering over the structure of the drug, which slightly improved the result. And then we added the two types of interactions. The first type of interaction make a lot of sense, but the second, this was a surprise. It really improved the results. And the last line, it makes no sense, because it means that the improvement from adding both types of interactions is so marginal that the drug-drug interactions are enough. No one talked about it in the literature, so we stopped and looked for the mistake. We visualized the data, we looked for the bug, but this was the result. Till now, we didn't find any mistake, and we published it, so hopefully there isn't a mistake. And we needed to find some explanation to what we see. So we can clearly tell that drug interactions, they are not a reason for anything. They are some mediator between the actual reason a drug is anti-cancer and the fact that it is anti-cancer. And we can see this mediator in the middle. And the thing is that when you think about it, drug interactions, it means that you take two drugs together as a person and something bad happens, a side effect. This is something that is very high quality, because as a drug goes to market, it's being prescribed to patients, people take it, and then they start complaining. And the authorities, they encourage the doctors and the patients to report. So you have a natural experience on humans. This is something you do not do in the laboratory. And this data is probably so high quality that it can really help mediating this activity. Then we had another neural network. This was a secondary one. Drug target on humans data was very, very small. And we saw that on yeast, it's much easier. There is a huge data set for yeasts, and we took one, we trained the same neural network for predicting between a molecule and a target, if it activates or not, or does nothing. And the last line here, this was very helpful because we used the opposite. Instead of nothing, we saw that there is something with pretty good accuracy. So this was another useful thing. And now, how do you continue? If you are a real data scientist, you'll say, take all of the drugs that are on the market, put them in the model, rank them according to the probability, and take the first one. But life is not that simple. We did this, but not all of the drugs are available to us. We cannot buy them. We're a small laboratory in Ben-Gurion University. You're starting getting questions. Why do you need this drug? What are you going to do with it? Send me all the results. Some compounds are very expensive, so we cannot even afford buying them. By the way, the molecules we worked with, they cost now something like $4,000 for a very small amount. So we cannot repeat that. And the domain expert had his opinion, which was very important, because they took active part here. And this system is not meant to replace anyone, but to help in the process. So we focused on three compounds from the tetracycline family. This is a family of antibiotics that is known to have anti-cancer activity, but no one ever talked about these three compounds. So we decided to talk about them. We took them to the laboratory and started with in vitro experiments. So you can see the line on the bottom. This is eravacycline, the antibiotic which was most interesting here. And you can see that on pancreatic cancer cells, very fatal cancer, it really reduces the viability of the cells. Now, this is not my specialty, so bear with me. I'll do my best. And we decided to move forward with the eravacycline. So we tested also the mechanism of action of this drug. And we had two targets that we were focusing on, POLQ and P53. Both of them are important in cancer for the duplication of the cells. And you can see a nice dose-response relation between eravacycline and the expression of these genes and proteins. And if you have weak heart, I'm sorry, close your eyes and ears for 1 minute. This is from the in vivo experiment where we used xenografts and tumors were injected to mice at day 0. And after around 10 days, we started giving 3 types of treatments. One is the control. Second one is gemcitabine, which is the standard of care for this type of cancer. And the third is eravacycline, which is the antibiotic that the machine learning model highlighted. And after 10 days, first dose, second, 12 seconds, 14, 16, 18, and then it stops. You can see the line on the bottom. This is eravacycline, so the volume of the tumor was smaller at the end than when we started, which is good, better than the standard of care, which is gemcitabine. And on the left, you see the actual tumors that were extracted after the trial was ended. So you can see that the tumors are smaller in the group that had the treatment, the eravacycline. And it was great, great result. We're happy. And then we asked, why stop here? Why just repurpose drugs? It's important, but we can also show that we can generate new drugs. So this is what Eyal is doing in his PhD, is continuing this work. So he took this model that we developed, and he's using reinforcement learning to generate new compounds. And we were able to actually take them and show them the results in the laboratory. And you can see, again, that there is some activity there on the left. And we're continuing with that. So I hope to be here next year and show you more interesting results. So I hope that now you're convinced that with the right data science skill set, with simple assumptions, you can solve real big world problems. Thank you.
Okay, so questions from the audience?
Just a general question. What was the size of the data set? And out of curiosity, are there good, like pre-trained graph neural networks nowadays for small molecules?
Yeah, thank you for the question. So I'll start with the pre-trained. There are pre-trained versions of this neural network. They did not help, so we did not use them. The size was about 200 drugs for the positive cases, and we labeled each one manually. And around 800 for the negative set. These are the amounts. Sorry?
Very small data sets.
Yes, it's relatively small, and we use, because of that reason, we used for validation scaffold split, so not just random split, and finally it worked.
I have one more, if permitted. So the same groups, they published recently a paper where they looked for explanations, like scaffolds that best explain the activity. Have you ever tried something like that?
Yeah, so we tried explaining the reason for a molecule to be anti-cancer. It's actually pretty simple. They take the molecule, they have this built in to the package, and they start removing parts from the molecule until they see a large reduction in the probability, and this is the explanation. So we did this, and we showed it to the pharmacologists, and they said, yeah, okay, that makes a lot of sense, but it's not really explaining the reason. So maybe it means that we should do more around this area. Yeah.
Thank you very much.
Thank you for the questions.
Okay, let's thank the speaker again, and thanks everyone for being here with us for this session. Bye.